welcome to my channel. So in today's session we are going to study everything about carbohydrates. So starting from the monosaccharides, disaccharides, and also we will go to the different details of the reactions of these carbohydrates. So let's start. What are carbohydrates? So actually carbohydrates are also known as sugar. So this is the common name of carbohydrates. They are found in all living organisms. So they are present in different forms in different organisms that we are going to study little bit later. They are essential to the very source of life. Basically they are very essential. They are the parts of very essential molecules. For example, DNA, RNA. In DNA there is ribose and deoxyribose sugar are present in the nucleic acids, and also they are required to sustain the life itself. For example, conversion of carbohydrates into usable biochemical energy, ATP, right? So generally the names of carbohydrates include starches, saccharides, polysaccharides, right? And the common formula for these carbohydrates is this one, CxH2O whole Y. So here you see there is carbon and water. Carbohydrate, they are the hydrates of carbon. Okay, here X and Y may or may not be equal, so they could be same or they could not be the same. Okay, and the value ranges from 3 to 12 or more. For example, in case of glucose, it is 6, right? C6H12O6, right? So it can be written as C6H2O whole 6. Now the classification of carbohydrates. So there are different types of classifications itself. Okay, so based on different properties, based on different criteria, right? The most useful classification scheme divides the carbohydrates into groups according to the number of individual simple sugar units. How many simple sugar units, how many monomers of these carbohydrates present within the molecule. Monosaccharides, disaccharides and polysaccharides. Okay, so monosaccharide, as the name indicates, mono means single, so it contains one sugar unit. For example, glucose, galactose, fructose, etc. Disaccharides, they contain two sugar units. Okay, di, two. In almost all cases, one of the sugar is glucose. Generally the one of the sugar is glucose and the other one may vary. Okay, so examples are like the other sugar could be galactose, fructose or another glucose. Common disaccharides are maltose, for example, okay, and lactose and sucrose. All of these we are going to study. Polysaccharides contain many sugar units in long polymer chains of many repeating units. So polysaccharide means multiple units. So there is also one term which I would like to introduce over here is oligosaccharide. Okay, oligosaccharide. So here oligo means less than 10 units. Okay, so less than 10 monomeric units are present, then it is oligosaccharide. Polysaccharide, it contains many sugar, more than 10. Okay, many sugar units in long polymer chains in the repeating units. Okay, and most commonly it is glucose. Common polysaccharides are starch, glycogen and cellulose, right? Now isomers. So basically these carbohydrates exist as isomers. Optically active isomers are also present, and the simplest, the basic, the most basic carbohydrate is what? Glyceraldehyde. Always remember, glyceraldehyde is the simplest carbohydrate. And how many number of carbons are present in it? There are 3 carbons present in it. Okay, so it exists in 2 mirror forms. Here you can see D form and L form, right? And the absolute configuration of this molecule is defined by the carbon which is far left as the D-glyceraldehyde. Okay, here right? The aldehyde group in the up direction, the OH group must project right side of the molecule. So here if it is on the right side, then it is upward, okay, and if it is left side, then it is downward. So like this we can designate each and every molecule, whether it is D molecule or L isomer, right? So monosaccharides are designed to the D family according to the projection of OH group over here. So here you can see clearly, like in the previous slide I have also indicated. Here OH group, if you see, this is the last OH group, okay, which is farthest from the carbonyl group, okay. So over here if you see, it is right side, okay. If it is on the right side, then it is D, okay, and carbon 1 contains carbonyl group, okay. So this is D isomer, okay, this is the D isomer, and this is the chiral carbon, and carbon number 6 is not chiral, so that is why we are not considering it. We will consider only carbon number 5, C5, because it is the last chiral center carbon. Now complexity, based on the complexity also they are defined as, they are classified as simple carbohydrates and complex carbohydrates, right? So simple carbohydrates, as we have already studied, monosaccharides, and complex carbohydrates include disaccharide, oligosaccharide and polysaccharides. Now on the basis of size also we can classify these carbohydrates. For example, if the number of carbon atoms present in the carbohydrate, it is 3, then it is called as C3 sugars or triose, okay. Example of triose we have already studied is glyceraldehyde, okay. So it is having 3 carbon atoms. Tetrose, C4 sugar, pentose, C5 sugars, 5 number of carbon atoms, hexose, C6 sugars, 6 number of carbon atoms, heptose, C7 sugars, and they contain 7 carbon atoms, right? So here are certain examples, certain representative molecules for each and every type of sugars, right? So again, based on the carbonyl functionality, whether it is aldehyde or whether it is a ketone. So if you remember, the aldehyde group is this one, okay, it is having H and R. So this is aldehyde. And in case of ketone, it has both the R groups attached to the carbonyl carbon. So this is ketone. So sugars having aldehyde groups are called as aldose sugar. Sugars having ketone functionalities, ketone group, are called as ketose sugars. Now coming to the reactivity. Okay, so there are reducing sugars and non-reducing sugar. Reducing sugars and non-reducing sugars, based on their reactivity, they are classified. So all those carbohydrates which have free aldehyde or ketonic group, they are called as reducing sugar. So they should have a free group. It should not be attached with any other bond. Okay, and this free aldehyde or ketonic group can reduce Fehling solution and Tollens reagent. So that is why they are called as reducing sugar. So always remember, these solutions, these reagents are actually oxidizing agents. So it is reducing them, right? Example is glucose. All monosaccharides have free aldehyde or ketone group, so that is why they all are reducing sugars. Now coming to the non-reducing sugars. Non-reducing sugars, here the aldehydic or ketonic group are bonded. They are bonded, they are not free. That is why they are called as non-reducing sugar. Example is sucrose. While others in which these functional groups are free, they are reducing sugars, right? So that is the difference between reducing and non-reducing sugar. Now coming to the important reactions of these carbohydrates. Starting with the oxidation, sugars may be classified as reducing or non-reducing, so they themselves get oxidized. If some reagent is reducing reagent, then it means that it is itself getting oxidized. Okay, so if sugar is oxidized by these reagents, then it is called as reducing sugar. Okay, so here Tollens reagents is there and Fallings reagent is there, right? And there is formation of silver mirror or the precipitation of cuprous oxide upon reaction with these reagents. Okay, so Tollens test is commonly done to detect the aldehyde functional. Among other reactions, for example, if you see ribose sugar, for example, ribose sugar is C5 sugar. Okay, so here 5 number of carbon atoms present in the presence of hydroxyl bromide. HOBr is called as hydroxyl bromide. Okay, when it reacts with this reagent, this is oxidizing agent, gets converted into ribonic acid. Over here if you see, there is a carboxylic group. So aldehyde group is converted into carboxylic group. So ribonic acid. In case of HNO3, which is nitric acid, it is very strong acid. It is very strong oxidizing agent also. Here ribose, both the terminal carbons get oxidized to the maximum and they get converted into carboxylic group. This is called as ribaric acid. Okay, so these two reactions are very important. You should remember that. Now coming to the reduction, they also get reduced. Okay, so common reducing agent is sodium borohydride reduction. So it is very good reducing agent. NaBH4, sodium borohydride is NaBH4. Okay, so it reduces the aldose sugar and makes the ends resulting into additol chain. So over here if you see, this is the sugar. Upon reduction with sodium borohydride, it gets converted into alcohol. So it is entirely made up of all the OH groups present on all the carbons. Okay, so that is how you can detect whether it is reduction or oxidation. Okay, so CHO group is getting converted into CH2OH. Alletol and galactitol form from reduction of allose and galactose. Okay, so these are the examples. So alletol and galactitol, right? So here if you see, this is the summary of all the reactions. So in case of HOBr oxidation, aldonic acid is formed. HNO3 oxidation, aldaric acid is formed. Ribaric acid, if you remember. Then with the NaBH4, sodium borohydride reduction, what is formed? Alditol. Okay. Now there are other reactions where there is chain shortening and chain lengthening. Okay, so starting with the chain shortening, example is rough degradation. Okay, so starting with CN aldose sugar, n number of carbon atoms. So you please give attention to it. So here n number of carbon atoms present. Here in the presence of bromine in water, HOBr hydroxyl bromide, it gets converted into aldonic acid. As you remember, it gets oxidized, and here this carboxylic group is present. Then in the presence of ferric ion and a peroxide and calcium carbonate, it gets converted into Cn-1 aldose sugar. Okay, so here you see there are how many carbon atoms? Cn-1, and one CO2 is removed. One CO2 is removed, and which CO2, which carbon it is? The one which had this carboxylic group. Okay, so one carbon dioxide molecule is removed. So that is why it becomes Cn-1. Now coming to the next reaction, which is chain lengthening. Yeah, chain lengthening means the number of carbon atoms are increasing in the chain. Right? So over here the example is Killiani-Fischer synthesis. So this this reaction, here first HCl is used, hydrogen cyanide, in the presence of acidic medium. Okay, so hydrogen cyanide is added and it will increase, because cyanide is there is extra carbon which will increase one carbon in the chain of this sugar, aldose sugar. Then what is formed? Aldonic acid, because there is carboxylic group. And then there is lactonization and reduction, and upon which there is formation of, because it needs to be reduced, because this is carboxylic group, it needs to be reduced to aldehyde. So this COOH has to be converted into CHO. Okay, so reduction is required, and upon reduction what is formed? A sugar which has Cn+1 carbon atoms. Okay, so this is the example of chain lengthening reaction, which is Killiani-Fischer synthesis. There is another degradation chain shortening reaction which is called as whole degradation. This is an alternative technique. Here hydroxylamine is used. This is hydroxylamine. Right? And here if you see, it started with the sugar which has aldehyde group, aldose sugar it is, and upon reaction with hydroxylamine this compound is formed, and with the heating with acetic anhydride. So this is acetic anhydride. What will happen? Water molecule is removed. So this H and this OH is removed, and there is CN formation over here. Okay, and now in the presence of sodium methoxide, what is formed? This aldehyde group is formed. So this CN is removed. Sodium, actually over here sodium cyanide is removed. Right? And there is formation of one less carbon. Okay, and this CHO group is resumed because of the sodium methoxide. So this is another example of degradation chain shortening, where hydroxylamine and acetic anhydride along with sodium methoxide is used. Now coming to the disaccharides. See, this is a common, just for the introduction I would say, this is glucose, another glucose. These two are monosaccharides. These two are reacting and forming glycosidic linkage. Which kind of reaction it is? It is condensation reaction. Okay, and there is glycosidic linkage formation, and there what is required? Acidic medium is required. So this is actually a disaccharide. Now starting with the maltose. So this was actually maltose, where two glucose units were reacting together, right? So this maltose occurs in the sprouting grains. How it is formed? By the partial hydrolysis of starch and glycogen. In the manufacture of beer, maltose is liberated by the action of malt. Which is, what is malt? Actually it is germinating barley. So while making of beer, alcoholic beverages, so it is produced from the barley, germinating barley, because there are enzymes present which will act upon it, on the starch, and will produce maltose. Maltose is about 30% as sweet as sucrose. Okay, so human body is not able to metabolize maltose or any other disaccharide directly from the diet, so they cannot metabolize it directly. Okay, and also these molecules are very large, they cannot simply pass the membrane. Okay, so that is why what is required, hydrolysis is required. So how it will happen, with the help of enzyme. Which enzyme? Maltase enzyme. So maltase enzyme will act on it, it is converted into glucose subunits and these are absorbed. It is a reducing sugar. Okay, and because the anomeric carbon, it has a free aldehydic group, it can open and form aldehydic group. Okay, so over here these units are joined head to tail fashion. So the glycosidic linkage is alpha glycosidic linkage. The OH group on the anomeric carbon of the second sugar can be in the alpha or beta position. Over here, if you see over here, see, this is beta orientation and this is alpha orientation. If it is below the plane of the ring, it is alpha, and if it is above the plane of the ring, it is beta. This is carbon 1. Over here it is associated with the glycosidic linkage, over here also, but this carbon, if you see, this carbon is free, it is not associated with the bond formation of another molecule. So this carbon, this anomeric carbon over here, is responsible for the reducing nature of maltose. Okay, now coming to the lactose. So in this video I will be covering disaccharides up till disaccharides, then in next video I will include polysaccharides. So lactose is also known as milk sugar. Human milk contains 7.5% of lactose. Cow's milk contains about 4.5%. This sugar is one of the lowest ranking in the sweetness, least sweet, 1/6 as sweet as sucrose. Lactose is produced commercially from whey. What is whey? It is actually a byproduct of byproduct during the manufacturing of cheese. It is very important as infant food because infants can digest it easily. It is a reducing sugar, again it is a reducing sugar, and it is made up of galactose and glucose, which are joined by beta 1,4-glycosidic bond. So beta means the carbon is above the plane of the ring. The two monosaccharides obtained from the lactose are by acid hydrolysis or by enzyme lactase. So with the help of enzyme or acid hydrolysis, it is converted into galactose and glucose. Over here you see beta 1,4-glycosidic. Beta, why beta? Because it is above the plane of the ring. Carbon 1 and carbon number 4. Right, and this is the second sugar. Right, over here this anomeric carbon is free. This anomeric carbon will cause the reducing nature of this sugar. Upon hydrolysis in the presence of lactase or in the presence of acid, the bond is cleaved, this beta 1,4-glycosidic linkage is cleaved, and what we will find, we will get galactose and glucose. In some cases there is deficiency of this enzyme lactase, so these individuals cannot digest lactose, okay, because they do not have the enzyme to cleave it. So that is why they cannot digest the milk, cannot digest the lactose present in the milk. Galactosemia. So it results from the absence of enzymes needed to convert galactose to glucose. So this is another condition. Okay, there is lactose intolerance, there is galactosemia, and certain bacteria can metabolize lactose, forming lactic acid as one of the products. Now coming to the questions. So this is a trisaccharide over here. So you have to tell whether each glycosidic linkage is alpha or beta. This one is alpha or beta and this one is alpha or beta. Over here the OH is above the plane of the ring, so this is beta, and over here it is below the ring, it is alpha. Okay, so beta and alpha. Now coming to the next disaccharide, which is sucrose. It is the largest selling pure organic compound. Beet sugar, cane sugar, table sugar, simple sugar. So all of these are the names. So various names are given to sucrose. They are obtained mainly from the sugar cane and sugar beets. The juices are 14 to 20% sucrose. Okay, so how it is obtained? So first juice is prepared, then from the juice it is evaporated, water is evaporated, then there is crystallization, there is dark brown liquid, okay, and which is remains of the juice, that is sold as molasses. In the sucrose, which type of glycosidic linkage present? Alpha 1 beta 2 glycosidic linkage, head to head linkage. Over here both the anomeric carbons are involved in the glycosidic bond formation. Over here this is anomeric carbon, again this is anomeric carbon, both are involved. So this is actually beta, this right, this is actually alpha, sorry, this is actually beta. Okay, so alpha 1 beta 2 linkage, alpha 1 beta 2 linkage is formed and one water molecule is removed. Which enzyme is required for the hydrolysis? Sucrase enzyme. It is also called as invertase enzyme. This invertase enzyme actually hydrolyzes this sugar into glucose and fructose. And this mixture is formed, this 1:1 mixture is referred to as invert sugar because it rotates the plane polarized side in the opposite direction as to that of sucrose. So sucrose is in one direction and this mixture which is obtained is actually rotating the plane polarized light into opposite direction. That is why it is inverting the polarization. Okay, so sucrose is readily recrystallizes from solution, but invert sugar has greater tendency to remain in the solution. So that is why it is more important, it is more applicable in the industries because it does not recrystallizes and it will remain in the solution form. So that is why we prefer invert sugar for the commercial products. For example, in the manufacturing of jelly, candy, in the canning fruits, the recrystallization of sugar is undesirable. It won't look good. The product will not look good. So what we do, we use invert sugar instead of proper sucrose. Okay, and also fructose is sweeter than sucrose. The hydrolysis add to the sweetening effect also. Okay. So this is the question. Here you will have to find out whether this sugar is having alpha or beta glycosidic linkage over here. So this one and this one. Over here if you see, both are head to head. This is head to head, this is head to tail. This is head to head. This is head to tail. This is alpha or beta, this is alpha 1, beta 1. Over here this is beta 1. This is not required because over here it is anomeric carbon this is C1, this is C4. So beta 1 glycosidic linkage, over here alpha 1 beta 1 glycosidic linkage is present. This is another example. Okay, so here you have to identify whether it is reducing or non-reducing sugar. Okay, so reducing sugar which has free aldehydic group. Okay, here both the anomeric carbons are involved in the bond formation, glycosidic bond formation. So this is non-reducing sugar. This is non-reducing sugar. Here this is C1, this is C1, this is C1, this is C1. So here only this one is involved in the bond formation, but this one is free. This aldehyde group is free. Okay, so when the ring will open, this will be free aldehyde group. It is not involved in glycosidic linkage, so that is why it is reducing sugar. So I hope this session, this video was useful to all of you. If you liked it, please like, share and subscribe to my channel, and I will keep on posting new videos, and I will cover all the topics, especially for all the exams, bio related exams. So thank you everyone for watching my video, and take care.